Okay, we are starting day 14. We're still here. Side quest 0. Today's gonna be interesting. Usually, as always. Live. Live. Chat. Chat. Okay. We're starting from a side quest today. Live. Chat. So we're going back to the cognitive Litecoin. We're starting from a side quest today.
I coined this thing called a cognitive Litecoin, which is basically the boundary.
Okay, so I bought a white.
I coined this thing called a cognitive Litecoin, which is basically the boundary of the self.
Okay, so I bought a.
I coined this thing called a cognitive Litecoin.
Oh, here we go. Here's a Michael Levin. Blocked. Only 42,000 subscribers? That's so strange. That's Xenobots. That's how they programmed Head or Tail. This is so cool. Subscribed.
I coined this thing called a cognitive Litecoin, which is basically the boundary of the self. It's meant to be the thing that distinguishes a self from the outside world. And what it is the scale of the largest goal you can pursue. So again,
yeah, the scale of the largest goal. That's exactly what SAP Cognitive Litecoin. We're just playing around. Cognitive Litecoin. Starts with a template. No. Create repository. The license MAT. What does the MAT license stands for? Anyone can use it? What does the MAT license stands for? Basically, what is the license that anyone can use the source code for commercial or regular work? Upgraded robot. Greater in name and memorable as how about upgraded robot. What's the what? Prompt. I don't need to prompt. I need to clone it. Copy. And we're gonna start from cd code git clone. Perfect. And we're gonna open it in Z. Oh, Cognitive Litecoin. Let's see, let's get started. Let's see what's up here. Interesting. I have no idea how to navigate this. Search. How can I start agent panel? Cloud engine. Come on, come on. And where we stopped it, it's oh, we did this, yeah. By the way, every idea when we have be validating it. It's good. Oh yes, that's what I like. Good luck. Before so, we are building something that does not exist. Yeah. Let's go. Upgrade command, screen. Yeah, it's my agent. I'm excited how it's gonna turn out. So thinking, that's good. That's probably oh, the files in the right side. Okay. We got this part. We can close it. Yeah, we read about that. I'm very excited. This laboratory representation. Osmosis, it's when water goes from a less concentration to from a big concentration of something to less concentration of something. Spontaneous net movement of water molecules through a semi-permeable membrane from a region of higher water concentration, lower solute concentration, to lower water concentration, higher solute concentration, aiming to equalize solute concentration on both sides. It is critical biological process. So if you put more solute, I'm assuming salt, something like similar to salt, right? Like potassium or yeah, whatever. It could be almost anything. But an example, if you put a lot of salt in one area A, and then no salt in area B, and put the water in area B, and connect those two environments, the water will go into stuff that it want to dissolve. Something like that. Are we running? Still running, that's good. It starts at 3 agents. We're gonna wipe code that stuff. That's gonna be so cool. And we are back. That was a side quest. We started from a side quest. So we back to our questions. Only 5 left. What is a plasmid, and why do scientists care about them? Oh, by the way. Right. That's a question number 3 out of 50. That's I have no idea what it is. This is probably question number 3 out of 50. That's I have no idea what it is. I couldn't even guess. Double check if I'm correct, and also give me the answer. Oh, by the way, what it's running? Opus or default recommended effort? I want to run Opus. I want to make sure I'm running Opus. Looking for threads from the external agents. Import threads from agents like load, import threads. Import threads from agents like, I don't know, like command enter. Oh, that's so cool. Kind of cool. But how can I switch to? Yeah, that's better. Let me count your true no idea answers. Osmosis. That's what I just described, right? Osmosis, yes, we can close this. 3D cognitive light color research, we can close this. Limits. Semantic versus germ cell, I don't know. So germ cells, that's basically dividing normally, and semantic, that's the one that's divided only with the half of chromosomes. I think my brain is getting stale. Like every cell in your body that is not involved in reproduction, yes, skin, muscles, neurons, liver, blood, all somatic. Or opposite. Somatic, it's divided normally. Soma, body. Okay, and the germ cells, the lineage that produce sperm and eggs, the ones that's for reproduction with the half. Wow, I remember it right. I just mix the terms. $45 plus mid. Let's see if I know that word.
Plasmid.
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 I have some questions in chat from anonymous user. Let me answer that. I just need to know where I can do that. Studio mode? No. It should be somewhere in... Oh. It should be somewhere in YouTube, I guess. I'm assuming. Where is that? Here we go. Come on. Okay. Let's install it. Later. Back. Building a web camera, stream software, close. So I'm going here, and here I see the chat. Hello. Okay. we are on why we I hard pivot from IT to biology. So answering initial 50 questions so AI can identify my gaps and we and build a for me. My first reply in comments, and thank you for your comment, anonymous user. That was side quest number 2, obviously, from our... Oh, by the way, we got a Twitter now. Finally, we got a Twitter. Oh, X. Where is my... side quest count? 2. Really quick. Oh, it's not gonna be really quick, but I'm gonna do it anyway. Front-end research synthesis, recommended React task white plus TypeScript, React 3 fibers. Okay, I'm okay with that. I'm not a strong front-end engineer. Tungsten query, cache, architecture sketch, top 10 Fox study ranked. Oh, we're gonna look at them, yeah. Prove selling. What is this? No, that's not it. Oh. That's going to be side quest number 3. I need to see live examples. Oh, yeah. That's what we're talking about. Can you zoom in? You can zoom in. Oh, yeah. Oh, here you go. It's so slow, however. Come on. It's incredibly slow. Risk register, nested counts, visual artifacts, highest R2, floating point, precision deep. However, complexity estimated animation strategy. That's good. Let's start from scratch. Let's pick your best recommendation for the front end, and let's also beat the backend microservice, and we can use a database. PostgreSQL to store data. Let's seed that data, and let's use backend microservice to take data from the database using Golang, my usual Fiber, Go Fiber API framework. Feel free to take a look into other projects inside of the code directory on my machine. Build the backend. Actually, don't even connect to the database. Just build the backend with MAC data, and also build a front end that will call that backend instance to get that MAC data, and we can start drawing. Later on, we'll deploy it. What's more important for me is that the backend with the data stays the same, and the way how we measure and size different species or agents would be calculated on a backend, and just give a flat numbers to the front end, and front end just build the visualization. We'll start from a first front end, and then we're gonna swap every single one based on your recommendation gradation and see which one applies for us the best. Okay. What is plasmid? A plasmid is a small circular loop of DNA that exists inside bacteria, separate from the main chromosome. Think of it as the bacteria chromosome as the main hard drive, and the plasmid is as a USB stick. By the way, where is my USB stick? I forgot it upstairs. It's extra, optional, independent DNA that replicates on its own. By the way, let me tell you what I was realizing over the weekend. I think my understanding of DNA, it's a little bit outdated. It used to be, right? I always thought, as probably majority of people, that DNA, that's what's dictate how you're gonna look and everything. But it's actually not at all, because like answering this question and realizing like majority of DNA and information that we have in DNA not used at all. So DNA, it's kind of internet of things, of knowledge, and then RNA actually turn off and on certain part of it, and that has been read. So in order to build our cells, we use only like what, 20%, maybe less, of DNA. Also, DNA doesn't dictate what organism you're gonna be. That's what blows my mind. So xenobots, it's the same DNA, but completely different organism, because it's in different environment, because the surrounding cells doesn't tell it what to be. This is so, because before I was writing simulation on cells, I always was use DNA as a core, but now DNA, it's not a core at all. It's like literally storage of everything, and you like pick and choose whatever you need, and only small part has been used. It's hard for me to formulate it in words, but the idea already brewing, right? So the way we are, it's not because of DNA at all. Like, not at all. And I really start thinking that, or not thinking, start leaning the theories that's saying that some other mechanism of dictating what it should become from the cells division as the embryo, but not in DNA. DNA is kind of like, here's the list how to build the basic tools or functions. How to build nerve cells if you need it, how to build the muscle cells, how to build, here's a library of all things that you possibly we can build using chemical and molecular structure, right? inside of the cell, and cell is the factories of building those. And we have a blueprints of pretty much any processes like electron transport chain. How to build that shit? It's written in DNA. Or any other processes, like how to build amino acids. We build in 11 of amino acids, and 9 of them we don't. And what's cool about it, that's before our ancestor was building it, the other one as well, but we decided to not use those tools anymore because they're too expensive to produce. Not the tools, those system of tools not actually selected and exist in our body. They still most likely exist in DNA, or maybe not. But we're not utilizing them because to build essentials like amino acids, it's way more expensive. 18 ATPs to build one of them. It's way more expensive than just take it from outside. Eat it, pretty much. And that's what's interesting. So DNA, it's not who we are at all. DNA, it's a big library of different blueprints that could be used to support the organism or different tissue and so on. And that's what did not click to me before. Now it's finally like, wow. Two things that influence of me radically changing my perspective on DNA. Number one, that we don't use 90% of DNA. I think that's... I'm gonna double check that. What is the percentage of DNA we are using? Is it 90% we not used at all to build our body and everything what's needed for our body, all our cells and so on, to build a human organism? And the information that's not used in DNA called... And what's the name of the information that's in DNA but not used to build our organism? I think retrovirus or something. And you're circling around the real concept, but the framing needs some correction. Let me let's school me. Number, the 1.5% number is real, but unused is wrong. Only about 2% of human DNA codes for proteins. These are exons, the segments that get transcribed in RNA and then translate into proteins. So in the narrow sense of builds proteins that constitute your body, yes, the vast majority of your genome doesn't. Oh, so it's only for the proteins. Interesting. But the second part was the xenobots, for sure. Xenobots was very interesting experiment. So let's correct this one. But that doesn't code for protein equal not used. A significant chunk of remaining 98% has regulatory roles. Promoters, enhancers, silencers, insulators that control when, where, and how much of each protein gets made. That I didn't know. Very interesting. That I definitely didn't know. In that case, it's not a library of blueprints. The blueprints only 2% of it. Everything else it's rule-based. Rules, right? And going to the code analogy, then DNA is obviously like kind of a hard drive, right? From where we read an instruction of each planet gets made, and that's definitely a side quest. But very interesting and necessary one. Think of it like the exons as the function bodies, but the regulatory DNA is the entire control panel. Not clicking. The orchestration layer that determines which functions get called in which cell type at which times. That's I got it. Without it, you'd have a pile of proteins coding genes with no coordination. Yeah, but how does it coordinate? So ribosomes are building proteins, right, from RNA. But how does it orchestrate? The term you're reaching for is endogenous retroviruses. Retroviruses, yes, and more broadly, transposable elements of retrotransposons. What the fuck? About 40%, I'm losing, like this is too much. About 40% of human genome is derived from transposable elements, sequences that can or historically could copy themselves and jump around the genome. The major categories that's the junk DNA debate is generally contested. Interesting. Encoded project in 2012 claim. That's a cool project. Encoded, they basically sequence everything, right? In one of the human. In 20 claimed 80% of the genome has biochemical function. Whoa. But many evolutionary biologists pushed back hard, arguing that detectable biochemical activity, like being transcribed at low levels, doesn't mean functional in any meaningful evolutional sense. Okay, so there's like nobody knows. It's still debates. The condensation is probably its substantial fraction of the genome really is non-functional remnant evolutionary debris, but we don't have precise number. It's like and as oversimplification. The key mental model correction: DNA isn't binary, use versus unused. It's more like a code base that has active record, infrastructure config, regular elements, deprecated but still reference model. That code, yeah, totally makes sense. That nobody cleaned up, and vendors copies of each ancient dependencies that got absorbed into the wrapper. That's your ERV. Yeah, the ratio of very good. Okay, so I understand that's only 2% goes into kind of blueprints of proteins. Super clear to me. Great, thank you for clarification. But how is this orchestration level / configuration level or function level, right? So when to call what and so on, how is that written to DNA, and how do we know? How does it works? And how do we know it works, and what experiments has been done to prove it? I didn't like I was not recording the voice, so I'm gonna repeat myself. Can you describe me a little bit better how do we know that information is written in DNA? And most importantly, how does it work? How's the orchestration level work in a cellular or on a cellular level or in the organism level? Basically, how does the organism know when to turn on certain enzyme or turn it off, or enhancer or promoter or silencer? When do they turn on and off? I thought the brain does it, but I'm assuming brain only controls information flows through the neurons and triggering the limbic system information, right? The one with I'm spacing out. The one they use hormones, yeah. So it's basically flooding a blood system with hormones. API web R3F. Why is R3F? Just put a web. This is the right question to ask next. It knows how to do compliments. And your intuition about hormones is particularly correct, but the full picture is more interesting. Yeah. Spill it. Than any single control system. Let me build it up layer by layer. Oh, we're going deep today. We're going deep. Hello, success. Hello, yep. Layer 1, the cell autonomous machinery, the base runtime. I love it. Every cell in your body has the same genome. True. Same source code. True. What makes a neuron different from a liver cell is which genes are expressed. True. And true, I mean, like, I understand this. It's not something new to me. This is controlled at the cell level by transcription factor, TFs, proteins that bind to specific DNA sequences to turn them on or off. Those promoters, enhancers, silencers I mentioned, and either recruit or block the transcription machinery that's bind to specific DNA sequences and either recruit or block the transport machinery. Okay, so TFs are promoters, enhancers, silencers. RNA polymers, the enzymes that through DNA interact, can't just land anywhere. It needs a landing pad. The promoter region, typically just upstream of a gene, but the promoter alone is often not enough. Enhancer, a DNA sequence that can be thousands of base pairs away from the gene they regulate. They're losing me. Transcription factor proteins bind to those enhancer and then intermediate loops around physically. So the enhancer bound TFs make direct protein. It explains me how. It doesn't explain who decide. Like, it doesn't explain decision process. Some works the same way, but so the like, chromatin state. The permission system. Oh, okay. DNA isn't floating around naked. It's wrapped around histone proteins, those circles, right? Forming chromatin. How tightly it's wrapped determines whether the genes are even accessible. Histones can be chemically modified. Blah, blah, blah. And these modifications either open up. DNA itself can also directly usually silence genes. This is not your permission. Local cell-to-cell signaling. Okay, that's better. Cells talk to the immediate neighbors through several mechanisms. Paracrine signaling. I think that's what YouTube video would be. Intercell signaling. Communication cell signaling pathways. Osmosis.org, I like that.
To make a multicellular organism, cells must be able to communicate with one another, and to do it, cells often send out tiny chemical signals that act on the receptors of other cells. Signals can be classified according to the distance between the signaling cell and the target cell. Autocrine signals are produced by a cell and go to its own receptors, so the cell sends a signal to itself. Paracrine signals are produced by a cell and go.
What is that? Cell signal to itself? Why? That's actually a very interesting example of recurrent function. This is so cool. Like, I can write cell simulation completely new way, knowing all this machinery. Very, very interesting. The cell signals itself. It's a recurrent function.
Go to target cells that are nearby, and endocrine signals are produced by a cell and go to target cells that are further away. Examples of these include hormones that are secreted into the bloodstream, as well as cytokines that can be released at the site of injury and act on the brain to cause a fever.
I need a screenshot of this.
Signaling molecules, or ligands, can be hydrophobic, meaning that they tend to repel water, or hydrophilic, meaning that they tend to stay in water. Hydrophobic signaling molecules can't freely float in the extracellular space, so they're brought into the target cells by carrier proteins. Hydrophobic molecules can diffuse across the cell membrane and bind to receptor proteins inside the target cell, either in the cytoplasm or in the nucleus. Most signal molecules are hydrophilic, so they can freely float in the extracellular space to reach the target cells, but are then unable to cross the cell membrane. So to pass on the signal, hydrophilic molecules bind in receptors on the cell surface. These receptors are transmembrane proteins with an extracellular end that binds to the ligand, and an intracellular end that triggers a signaling pathway inside the cell. We can think of the cell signaling pathway in 3 stages. The first stage is reception, which is when the target cell's receptors bind to a ligand. It's like a key fitting into a lock. Then there's transduction, which means that the receptor protein changes in some way, and that activates intracellular molecules.
That's interesting too. Yeah, I know part of it.
The second messengers. The third stage is the cell's response to the signal. Zooming into these transmembrane receptors, there are 3 major classes: G protein coupled receptors, enzyme coupled receptors, and ion channel receptors. Enzyme coupled receptors and ion channel receptors.
I need another screenshot, and this is definitely side quest number 4.
G protein coupled receptors are 7-pass transmembrane receptors. These are like really long proteins that have one end that sits outside the cell and binds.
It's going deeper. That's not okay. I'm gonna go back into this endocrine system. What you are reaching for? This system you are trying to name, the endocrine system, use hormones. I think I called it the endocrine system, right? At the end, secreted into bloodstream by glands, thyroid, adrenal, blah 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 blah. You're right, that's floods the blood system. Hormones are broadcast signal in a receptor, then the mechanical inclusion. I was asking not that question. I was asking is fast build. So how does the organism know when to turn on turn gene on? It doesn't know in any centralized way. It's a convergence of multiple signal type at each individual level. The cell own internal state, which TF it's already expressed. Its chromatin configuration is development history. Determines the baseline. Hormones in the blood provides organism level context via stress, growing, reproducing. Local signals from neighboring cells provide tissue level context, and the nervous system modulates the hormonal landscape in response to external condition. Okay, to loop back, DNA stores about 2% of instruction how to build the proteins. That's given. Then you say 98% it stores information about regulation of the different processes. When to start, when to stop, what to turn on and what to turn off. But then you explain that regulation actually it's real-time data coming from neighboring cells, nervous system, hormone endocrine system. So if regulation it's just accumulation of different signal and reacting to those, that means it does not store in DNA. So try to explain me again from a different angle what those 98% of code base, aka code base, is used for. Let's see if it understands me. Oh my god, that was slap. That was the AI slap. We caught AI slap in this. Like, how can you study when there's always AI slap? My first answer and your logic is exposing the slappiness. Called me the slappiness exposer. That's my new Twitter handle. Let me be honest about what actually going on. You're right that the regulatory decisions aren't stored in DNA. Oh my god, you lied to me in the face. What is stored in DNA is the response logic, the handler function. Oh, so when we have too much adrenaline, do that. When we have too little of something else, do this. Now it makes sense. An enhancer sequence doesn't store turn on gene X at 3 p.m. on Tuesday. Exactly, that's what I was trying to point out. It stores something more like this binding site shaped such that transcription factor Y can dock here. That's it. It's physical shape, a specific nucleoid sequence that a specific protein can recognize and grab onto. The enhancer is the receptor, not the signal. Very interesting. I have another question. Okay, this is much better explanation. Thank you. And I'm glad you caught your own slap, or I caught your AI slap. Be very careful next time. Double check everything with the websites and citation. Don't just generate the answer. So my next question for this, let's say we receive some signal inside of the cell. It doesn't matter from what system, endocrine or from other cells trying to tell us, neighboring cells trying to tell, even from ourselves to tell itself. We receive the signal that we need to create let's say we receive a signal that we need to create more enhancers, basically more receptors of shape Z. And we turn on machinery, we go into the DNA, reading instruction, what needed to create a Z-shape enhancer. We passing it, or we reading that chunk of DNA, converting it into RNA, and then building that receptors. So those receptors, I'm assuming, builds on a cell membrane. Those enhancers or receptors get built on a cell membrane. Let's say we need to more of them, right? So it's more sensitive. But then how is homeostasis happen? How are we like turn off that loop? Like at what point we saying like, hey, this is enough of those? And also, do they die off eventually? Or if we build, let's say, 40 Z-shape receptors, it stays with the cell to the rest of its lifetime. That's what's very interesting to me. Those receptors that's built in membrane of cell, are they permanent or have lifespan? And who regulate when we need to decrease? So you basically explain how to increase them by reacting on a signal, and we build them from DNA instruction. But how can we decrease them? Do we physically dismantle them? Is there a mechanism for that? That's an interesting question. Good question. I'll get to turn over mechanism, but first I need to catch a confliction before it become a permanent misconception. Enhancers are not receptors. These are completely different things. Oh my god, why you were telling me that then? Why can Doug here? This is a physical shape, a specific nucleotide sequence that a specific protein can recognize and grab onto. The enhancer is a receptor, not a signal. I'm so confused. I don't know where to believe you, like when to trust you or not. You told me in the plain text the enhancer is the receptor, not a signal. I'm not sure if now I need to even read all this because we got here, so the regular DNA encodes, but here's where I owe you a correction. It's not mostly a regulation. I'll let you walk away with the impression that the non-coordinated is the orchestration layer. That's misleading. You told me that. Here's more harness decomposition of what's actually in that 98%. Genuine regulation sequences, enhancers, promoters, silencers. The total genome. This is the real control plane and it's generally important, but it's not 98%. Okay. Interest takes up roughly 25% of genome. These are sequences within genes that get transcribed into RNA, but they're spliced out before translation. Some contain regulatory elements. Oh, this is so... We're going deeper, but I don't know is it truthful path or not. Non-coding RNA genes according transcribed. So honestly, answer what is in for. A lot of probably isn't for anything. Again, we're going back to like it's not for anything. C-valve paradox. I'm not even gonna read that. An enhancer is a stretch of DNA. It's part of the genome itself. It doesn't get built or insert into membrane. I never told you. I told you receptor built from enhancer. It sits there in chromosome permanently as a docking site for transcription factor protein. You can make more enhancers. I never told you that. The way you make more receptors. They are part of source code, not runtime objects. Yeah, I know that. Receptors are proteins. Gene products. What? It's it the in chromosome permanently. So hold on. Enhancer. That's a stretch. Enhancer is a stretch of DNA. Receptor. That's receptors are proteins. Gene products. Yes, that's true, and that's exactly what I was thinking. They get transcribed from DNA, translated from mRNA, folded. I was explaining that, like, it couldn't understand my voice to text. That's what it is. Folded and then typically trafficked to the cell membrane. Okay, we getting there. Or sometimes kept intracellular, as with steroid hormone receptor. These are things your question is really about. Runtime objects that gets instantiated, do their job, and eventually get cleaned up. So with that straight, your actual question is actually how do receptors and proteins generally get removed, and how does the cell avoid runaway positive feedback loop? Proteins are not permanent. Almost nothing in cell is. Oh, it's decayed. This is so cool. It's decaying. Yes. This is genius. How I was not thinking about it. I lost it. You actually hold on. Oh, allow dashboard go. Yes. Oh, by the way, I need to see, like, when you know what the context is, it's way easier to make a quick decisions. Most membrane receptors have half. Okay, yeah, that might be. Proteins are not permanent, and almost nothing in cell is. This might be the most important conceptual update. The cell is not a static structure; it's a system in continuous turnover. Most membrane receptors have half-lives measured in hours to tens of hours. Oh, that's so understandable now. Some last days. Very few last the lifetime of cells. The cell is constantly synthesizing new proteins and degrading old one. I'm wondering how. And the balance between synthesis rate and degradation rate determines how many copies of any given protein are presented at any moment. It's a steady state system, not a stockpile. How receptors get physically removed? There are two main systems. The first is the receptor-mediated endocytosis. This is mitosis, this is meiosis, endocytosis. I need to remove that word for my song. I didn't spell it correctly on purpose because I don't know how, and it's just a reminder. And this is elegant. The first is receptor mechanism, and this is elegant. Like, there is comment on YouTube that this is elegant, or like, how do you know this is elegant? 2 minutes, let's go.
What is receptor-mediated endocytosis? The following animation will describe the process of receptor-mediated endocytosis. To show what happens during receptor-mediated endocytosis, first let's draw our cell membrane. Note that the cytoplasm is on the bottom and the extracellular fluid is on the top. This means that the inside of the cell is on the bottom and everything outside of the cell is on the top of the page. In the extracellular fluid, there are solutes and particles which are floating around the fluid. These are called ligands. The idea behind receptor-mediated phagocytosis is that there are receptors on the surface of the cell which are specific for certain types of particles. This means that they will only attach to certain particles and leave the other particles alone. When a particle is detected in the extracellular fluid, the ligand attaches to the receptor. The receptor then migrates to an area which is coated in a bristle-like substance called clarithin. Clarithin helps the cell's own membrane form a budding pouch, also known as a vesicle. The vesicle containing the particles are transported to the inside of the cell for processing, while the clarithin and receptors are recycled and take their place back on the membrane of the cell, ready for the next round of receptor-mediated endocytosis.
What did I just watch? Like, I had no idea. I really thought those receptors just channels for proteins to attach and then pass through into the cell, inside of the cell. But I had no idea that they actually bound as a duck and crane, let's say, put this way, right? And then moved with the receptor itself into the cell, then getting released, and cell and receptor going back to the membrane.
Thanks for watching.
This is so cool.
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated endocytosis. Instead of circulating freely, cholesterol molecules are derivatized.
So this is an example of cholesterol.
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated endocytosis.
So into which cells? The ones that's inside of the bloodstream? Which one exactly?
Instead of circulating freely, cholesterol molecules are derivatized and packed inside
Derivatized.
low-density lipoprotein particles, or LDLs.
This is LDL. So it's kind of a cage for chickens. And cholesterol is the chickens in that case.
A protein and phospholipid layer surrounds the cholesterol molecules. The protein portion is recognized by LDL receptors on the surface of cells. An adapter molecule, called adaptin, binds to the tail of the LDL receptor that protrudes into the cytosol. Adaptin recruits clathrin molecules, which start coating the membrane. Assembly of the clathrin coat causes the membrane to bend and invaginate, forming a vesicle that buds off inside the cell, taking with it LDL receptors and the LDL particles bound to them. Once inside the cell, the vesicle uncoats and fuses with the endosome, the intracellular compartment that first receives all endocytosed material. The endosome has low internal pH, which causes the LDL receptors to release their cargo. Empty LDL receptors are recycled to the plasma membrane in vesicles that bud off from the endosome. Each LDL receptor makes a round trip from the plasma membrane to the endosome and back every 10 minutes. Meanwhile, the LDL particles need to be disassembled. The endosomal content is delivered to a lysosome, which contains hydrolytic enzymes that can digest the particles. Free cholesterol is liberated, together with amino acids and small peptides generated by digestion of LDL proteins. The cholesterol is then released into the cytosol to be used in the synthesis of new membranes.
Okay, the last part I don't know how, like enzyme breaking them totally makes sense, but how do they release it to the cell? Just like magic?
into the cytosol to be used in the synthesis of new membranes.
This is the most informative video that I watched like in a whole week. It literally blew my mind. I learned at least 4 mechanisms from it. Can I create a GIF? It's the material, I don't want to install it. I want to reshare.
And enters cells by receptor-mediated endocytosis. Instead of circulating freely, cholesterol molecules are derivatized and
Yeah, it's a chicken in a cage. So cool.
packed inside low-density lipoprotein particles, or LDLs. A protein and phospholipid layer surrounds the cholesterol molecules. The protein portion is recognized
Now I'm wondering who built that LDL protein.
by LDL receptors on the surface of cells. An adapter molecule, called adaptin, binds to the tail of the LDL receptor that protrudes into the cytosol. Adaptin recruits clathrin molecules, which start coating the membrane. Assembly of the clathrin coat causes the membrane
I need to check the grammar. I'm talking about how the cell in a microbiology cell, it's actually creating proteins receptors from enhancers. So it's reading enhancers instructions from DNA enhancers, then create a protein receptors, then transport them to the cell membrane. Then cell membrane actually use those protein receptors to bind whatever what's outside of cell, and use bunch of 2 other things that bind in the inner cell protein part, receptor part, to bend and bring that receptors with attached proteins into the cell, move it to the special area where enzyme break those protein down, and then those smaller parts can go into inner cell. So I want to leave a comment that I have no idea how of this amazing mechanism of the cell trying to move elements, meaning proteins, between different mediums. And different mediums, I mean outer cell environment, then low pH environment inside of the cell where enzyme break it down, and then the cell cytoplasm itself. And that, my friends, side quest number 6. We're beating records today. I really thought that's just a protein or receptor. It's kind of allow, aka tunnel, from outer cell into inner cell, and the protein pass through. That's what my understanding. But the interesting part, most likely, is that the also correct. It's just different type of mechanisms how outer elements from outer cell getting into the cell. It's just different mechanisms. Cell actually use both approaches, depends on what they need to bring. What you are describing by tunnel is a channel protein or transporter. This works small molecules and ions like sodium, potassium, glucose. Oh. Yeah. The protein spans the membrane and creates a pore that lets specific small things pass straight through. But what you were describing earlier, that's a receptor-mediated endocytosis. Endocytosis. Ion can transfer or enzyme. So you ultimately, and there is a third mechanism, right? How to transfer elements from outer cell into inner cell. If yes, tell me what it is, and then give me kind of a tweet so I can post on X. I need to build the agent. For the X. This is so cool. So this is three stages and 3 types. Ion channel receptors. That totally makes sense now. Enzyme couple receptors and G proteins couple receptors. So I know this and this. It's gonna tell me enzyme couple receptors. I'm gonna bet that. Yes, there is actually a third measure and a few more beyond that. The one you probably missing is phagocytosis, literally cell eating it. No. Instead of using receptors to pull specific molecules, the cell detects something large like a bacterium, or that cell extends its membrane outer wall. No. So the three big one is channel, transporters, tunnel, receptors mediate endocytosis, targeted grab and wrap, and factors the full engulfment. There's also simple diffusion. Small nonpolar molecules like oxygen just slip through the membrane on their own, no protein needed at all. And phagocytosis, cell drinking, gulping in fluid and whatever dissolves in it non-selectively. Give me the shortcomings that I was blown away by receptor-mediated endocytosis.
To bend and invaginate LDL proteins. The cholesterol is then released into the cytosol to be used cytosol. From the plasma membrane to the endosome and back every 10 minutes. Each brain empty others to release their cargo. Cellular compartment that first receives all endocytosed material. The endosome has low
It does all that's what's inside of the cell, right? Cytosol.
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 This is a brilliant question. I'm gonna post it on YouTube. Copy. Who is here from page 149 of biology textbook? Excellent power. Thanks. Best video ever. 100%.
Low internal pH.
Okay, we passed that. I should not. Was this? Was it? Oh, we answered question number 1. No way! We spent 90 minutes without even reading the answer. Because I went on a side tangent that's DNA, it's not as... Now we have to finish it. You're right to call this out, and I own you a clear accounting of what happened. I did write that word for word. The enhancer is the receptor, not the signal. And the one message later I told you enhancer another and corrected you for conflating them. That's me contradicting myself and then blaming you. What's this? That's not acceptable, especially when you explicitly ask me to be careful. Oh. That's actually a tweet. Today is a social media tweet. How do I do? I click post. I sent in screenshot. Is the emoji here? Okay, I need to pick up a daughter, but I gotta go. But that was fun. That was a really interesting. What I didn't know, what actually true. An enhancer is DNA sequence, a receptor is a protein. These are different categories of biological objects and chromosome. Yeah. What I did wrong, the rule going forward. I should not. I'm sorry. Okay. So let's answer the initial question. We have 2% of instruction for proteins. Then we have 25% instruction for enhancers. What else is there? That's so cool. This video is solid. Quick query first. The 20 is an enhancers. In my earlier breakdowns, the 25 was introns, which are different things. Let me reduce this clearly as a full decomposition. Protein coding sequences, exons. The actual instruction for building proteins. This is the part that gets transcribed into mRNA and translated into amino acid chain. Yes. 24 introns. These are sequences inside gene sitting between the exons. Sequences inside gene sitting between exons. A gene gets transcribed into RNA as one continuous stretch. Exons, introns together, and introns together. And then the introns get physically cut out by a molecular machine called the spliceome. The one, I think they use that for the CRISPR, right? Before the mRNA is sent to the ribosome for translation. Why do they exist? Some contain regulatory elements or produce functional non-coding RNA, but larger stretches appear to be tolerated filler than evolution never removed because there was no strong selective pressure to do so. What? I think we're going deeper. Regulatory sequence enhancers, 5-10% enhancers, promoter silencers, installations. Enhancers. This is interesting. So, non-coding RNA genes. We're gonna go back to that. That was fun.